Hi everyone and welcome to GCSE Revision with MFS. In this video we look at microscopy. We focus on the key parts on the specification and how you can gain full marks on exam questions. Please ensure you hit the subscribe button and the notification bell to ensure you never miss a video from GCSE Revision with MFS. If you would like to join our live lessons, the link to book your place is in the description below. Okay, we will make a start now. So welcome everyone to our GCSE Biology session. In today's lesson, we are going to be focusing on microscopy. Okay. Just before we get started though, it would be brilliant if you could all scan the QR code on the screen now to confirm your attendance to the session. Just so you don't get an email later on saying you have missed the session. So please, can you scan the QR code and just confirm your attendance to the session, and then we will begin. Okay, so it's just a quick form which asks for your name and email address, just so you can confirm you have attended the session. So I'll just leave that on the screen for a quick minute, and then we will make a start on microscopy. Okay, I'll just keep it on another 30 seconds for anyone who hasn't registered their attendance. It's just a quick form which you can scan to confirm your attendance to the session, just so you don't get an email that you have missed the session. Okay. If you have any questions as well, just let us know in the Q&A. We're going to begin with a quick sort of recap of what we did in our last lesson. Okay, I will pop the QR code back on towards the end of the session if you haven't scanned it already. Okay, so we will make a start now. So just a recap of what we did in our last lesson. So we were looking at prokaryotic and eukaryotic cells. Okay, and here is an exam question we had a look at in our last lesson. Just to refresh your memories, I've popped it on again, and then we'll have a look at what the marking scheme requires for this question. Okay, so just have a quick go at the question, and I'll just go through it in 30 seconds. Okay, so it just says give two similarities between the prokaryotic cell and eukaryotic cells in figure 5. Okay, so hopefully you've had a go at it. And if you can type your answers in the Q&A as well, and I can have a look at them and give you a mark on that as well. So give two similarities between prokaryotic and eukaryotic cells in figure 5. So I've actually put the marking scheme up on here. Okay, so this is what the marking scheme requires. So both of them have a cytoplasm, both of them have a cellular membrane, and both of them contain genetic material or DNA. They both also have ribosomes. So if we have a look at this sort of image over here, we can see we have a bacterial cell, liver cell, and mesophyll cell. We know that a mesophyll cell is a plant cell and the liver cell is an animal cell. And because of that, what we can depict is that both of them are eukaryotic cells. Okay, because both of them are eukaryotic cells, we know that they must contain the genetic material in the nucleus. Okay, and by that we can now depict from that the because we're comparing eukaryotic and prokaryotic cells, we can see the sort of differences there and the similarities. So we've got the ribosomes for protein synthesis. We have the DNA or genetic material. You cannot say nucleus because bacterial cells do not have a nucleus. You can say the cell membrane. You can't say cell wall because animal cells have a cell wall. Animal cells don't have a cell wall, sorry, and you cannot say anything along the lines mitochondria or anything like that. Okay, so be strict with yourself. Only allow yourself what is present on the mark scheme. Okay, so you can only have cytoplasm, cell membrane, DNA, genetic material, or ribosomes. Okay, and you can't have a process. As it says in the mark scheme, there is no other mark allowed it for cell subcellular processes. You cannot gain 2 marks for a subcellular process. Okay. Okay, just to let you know, guys, I've posted the link to registering to our session in the Q&A. So just to uh, confirm that you're here and present in the session, I pasted a link there as well. So make sure you fill that form out, as you don't want to be getting an email saying you've not attended the session. So just fill that form out and then you can carry on with the session. Okay, we have another just quick recap question over here. Before we move on to microscopy, give 3 differences between the prokaryotic cell and the eukaryotic cells in figure 5. So as we've just seen, we've just looked at the similarities. Now you need to look at the differences between the prokaryotic and eukaryotic cells. So we've just looked at the similarities as you have there. Now you just need to state the differences. Okay. So in the Q&A, have a go at it, just 30 seconds. We know the similarities, so what are the differences? Okay, so hopefully you've had a go at it. Here's what the mock theme says. So it says differences. So this could be structural or given the information in the diagram. So prokaryotic cell is smaller. Prokaryotic cells have no mitochondria. Prokaryotic cell has no nucleus, or DNA is free in the cytoplasm, or genetic material is free in the cytoplasm. Anything along the lines of that. If none of these are mentioned, you can state that prokaryotic cells has no membrane-bound organelles. Okay, but it has to be genetic material. Ignore genetic information, because if you say genetic information, you're not explicitly stating that you're referring to the DNA inside of the nucleus, or the one which is responsible for the characteristics. So make sure you're stating genetic material. Prokaryotic cell has a single loop of DNA, or prokaryotic cell has a single loop of genetic material. Again, it says ignore genetic information, and prokaryotic cell has plasmids. Okay, so again, you can't say circular rings of DNA. You need a name for them. They are called plasmids. And you can also have prokaryotic cells have smaller ribosomes, but that's not explicitly covered on the specification. They do have smaller ribosomes known as 70S ribosomes. You won't need to know that. You would just need to know that prokaryotic cells have smaller ribosomes than eukaryotic cells. Okay, so that's just a recap of what we did in our last lesson. Now, over here, I've got an image of a microscope, and this is a light microscope. Okay, this is how you would, this is the type of microscope you would use in a school. Okay, sorry about that. Okay, so this is the sort of microscope you would use in a school. Okay, we've got the eyepiece lens at the top, and that's the lens you would look through. Okay, you also have the objective lens that's used for magnification. So the two main things which are used to magnify your specimen, by specimen we are referring to the object you are looking at, the animal cell or plant cell, wherever you're looking at, that's known as the specimen. The eyepiece and the objective lens, they are used to magnify your sort of object that you're looking at, so we can get a clear magnified view of it. Okay, so that's the purpose of those two appliances on the microscope there. Okay, we've got the coarse focusing and fine focusing knob. What they do, their purpose is to, sorry, I will on mute there. So what we're going to do is we're going to get the look through the eyepiece lens. Sorry, it's a bit of technology going a bit wrong here. Okay, so we've got the, we're looking through the eyepiece lens. Okay, so we're looking through the eyepiece lens, and we're using the objective lens to magnify the objects. Okay, the focusing knobs, well, the coarse focusing knobs roughly focuses image, and the fine focusing knob helps you get a clear view of the image. Okay, So the purpose of the coarse focusing knob is to get an image which is roughly in focus. And the fine focusing knob refines that focused image. Okay, that's the difference. The fine focusing knob is normally the smaller dial on the stage, on the arm of the microscope, and the coarse focusing knob is normally the bigger dial on the arm of the microscope. Okay, the stage is where the specimen is placed. Over here, you've got the specimen. The specimen is normally placed on a slide. So we've got a slide and we've got our specimen. So what we're going to be looking at in the middle, and then this is placed on the stage clip on the stage. Okay, and then we adjust the magnification we would like to see it at, and we look through the eyepiece lens and we look at it through the magnification. Okay, so we've got, this is what we call the specimen. Okay, the specimen is what you're actually looking at, and this is placed on the stage. Okay, that is placed on the stage. Okay, and then you adjust your focusing knobs and objective lens just till you get a clear image of what you're actually looking at. Okay, so first of all, let's say we normally have three types of magnifications. We normally have times 10, we have times 40, and we have times 100. Those are the three magnifications we normally have. Let's say we're starting with times 10 magnification. Okay, we would use the smallest objective lens, looking at the specimen, place the specimen on, put the stage clips on so it stays in place, turn on the microscope, and you can look through the eyepiece lens, and then you can turn the coarse and fine focusing knobs until you get a clear image of your specimen. Okay, so just have a look at that and get yourself used to using the microscope. The purpose of the mirror is to reflect the light onto the condenser so we can see the specimen. So it gets the light, it attracts the light so we can view it under our specimen. Okay, so hopefully you visualize that. So you look through the eyepiece lens, you look at your specimen, that's where your cells are on. Okay, so that's where your cells are present. We've got our cells present there on the specimen, and you place it onto your stage, and then you attach the stage clips just to keep it in position. And then you start off with the lowest powered objective lens, so times 10 magnification, and then you gradually increase. So first view it at the lowest powered objective lens, looking down on the stage, and then you can focus the knobs until you have an image which is roughly in focus, and then use the fine focusing knob to get a clear view of your specimen. Then what you can do is move to the next magnification, which is times 40 in this case, and view it in a bit more detail and focus it until you get a clear image on the specimen. And then you can finally move to the highest magnification, times 100, and there you should be able to get a clear view of the cells, layer of cells in your image. Okay. Now, finally, before we end off for today, there's another point on the spec that I need to cover, and that's the equation for magnification. Okay, so we know how to use a line microscope. You also need to know the equation for magnification. Okay, now the best way to remember this is I am. Okay, just remember this, I am, and you will know your equation. What that means is that image size, so you've got the image size, yeah, is the actual size multiplied by the magnification. Okay, so just remember that image size is the actual size multiplied by magnification. Just remember I am, and that's it. Magnification has no units. Say something has a magnification of 100, you would just say 100 times. That's the magnification. Okay, now in the next lesson, what we're going to have a look at is we're going to have a look at comparing between the electron and light microscope. We will also be looking at unit conversion. So you need to know micrometers, nanometers, the conversions between them. That's a really common exam question as well. And we will be looking at some microscopy exam questions as well. So the purpose of today's lesson was just to get you a bit familiar with the microscope and give you the equation of working out the microscope. Okay, now just remember I am, that's how you will remember the magnification. You don't get this formula on the exam, so make sure you do learn it. It's called I am, really easy formula to remember if you remember it the way I have. Just I am. Okay, just fix that into your head and you will know it. Magnification has no units as well. Now, before we close off, I would like to give you the definition of magnification. Magnification, it's the amount an image has increased in size from the actual size to the image size. Okay, so the amount of times the image is bigger from the actual to the image size. Okay, and that's magnification. We will recap that in our next lesson along with resolution, which is another key definition you need to know. Okay, if you have any questions, guys, you can just let me know in the Q&A. This was just a quick sort of lesson guiding you on magnification. Okay, what I am going to do is I am going to put back this QR code just if you guys didn't confirm towards the start of the session, you will be able to confirm now. Okay, you can scan the QR code or you have the link in the Q&A to confirm your attendance. Okay, so what we've covered today is the process of microscopy, how you can use a microscope to magnify an object, and we've also had a look at the equation. Okay, and that's brought us nicely to the end of the lesson. If you have any questions or any worries or anything you would like me to go through, please do let me know in the Q&A. I'm here to help. So if you do have anything you would like to go through, just let me know in the Q&A. Okay, before you guys do go though, I'm gonna paste a link to buy me a coffee in the Q&A. And it's just a site we use if you want to thank us or support us for producing these sessions. What you can do is you can buy the coffee as a way of saying thank you. Okay. So I am going to paste the link in the Q&A. Also, if you are struggling with some topics with the GCSE curriculum, we have our own YouTube channel if you've not visited that yet. I will paste the link in the Q&A. So I pasted the link to buy me a coffee if you want to support us or thank us for producing the sessions. I will also paste my link to the YouTube channel, which has very educational, helpful videos, which can help you with your GCSE studies in the Q&A as well. Okay, if you would like to find that helpful. But if you do have any questions, please do let me know.